Biomolecular. Okay, so we will start. Okay. So good morning, afternoon, evening, hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us the 22nd seminar, and I call NSF Day. This year, everyone is talking about diversity, equity, inclusion, and accessibility. Also, I call DEIA. However, discussing the benefits and drawbacks of DEIA would make little sense because DEIA should be considered to ensure the basic human right, and DEIA must be the guiding principle of research activities and education, not a discussion point of benefits or drawbacks. DEIA is not achieved overnight. The cultural issues are too deep to solve them quickly. Leaders in academia and many sectors are talking about those issues while discriminating people in real life, and all rules on discrimination and harassment reflect the challenging situation that we have faced for centuries. If we had no issue, why should we have all the rules? That's my question. And hiring female researchers and supporting people of colors would not be sufficient because many leaders are still discriminating young people and minority groups without power, and these weak people are afraid of being fired and getting less support if they speak up. While watching Russian attack yesterday, I once again realized the importance of leaders, and such madness for considering only their selfish benefits should be stopped. And cultural change would be only possible by bottom-up approach taken by people together. We can change the culture together by giving our time, resources, attention, support, compassion, kind words, empathy, forgiveness, and of course, our love to everybody. And we rely on young people and governmental support to make this change really happen. So let's work together, and it will be so challenging, but God bless all of us and love us, and we can do it together. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Dave Rockcliffe, an NSF program director and cross leader in systems and synthetic biology. As you know, NSF is supporting synthetic biologists, especially young people with diverse backgrounds. And he is also a big supporter of our synthetic biology community. And what else do you want to know about him? So Dave, the virtual podium is all yours, and thank you so much for your time while you have lots of other duty today. Thank you so much.
Well, thank you, Tae-suk. It is a pleasure and an honor for me to be here talking in this kind of forum. And I must say a special thanks to you for organizing this effort to make such a global connection in synthetic biology. We are currently living in somewhat of a crazy time with all that's happening politically, socially, and economically. But I must say that despite all of this, it is an extraordinarily exciting time to be involved in research in synthetic biology, considering its seemingly endless scope and potential. This is the moment when we are defining our future with an emphasis on sustainability through a sustainable technology, and there is no better medium through which to achieve our goals in that way than with engineering biology. And I'm heartened to be part of this community that is seriously confronting the challenges of developing synthetic biology to a viable technology. I am a program director at NSF, as Tesa had just mentioned. And when I joined NSF at first, I was in the molecular biophysics program in the Division of Molecular and Cellular Biosciences. And after that, I joined the Chemistry of Life Processes program, which is in the Division of Chemistry. And that's a program whose awards are mainly in biochemistry and chemical biology. Therefore, synthetic biology was a new frontier for me. And since I joined this Systems and Synthetic Biology program 5 years ago, my fascination with the science has not abated to date. But I'll just say a little about NSF, right? And hope that you can find some kind of parting wisdom in what I have to say. But Systems and Synthetic Biology is not the only program at NSF with an interest in synthetic biology. Because there are synthetic biology programs that are housed all over NSF in the directorates for engineering and in the directorates for mathematical and physical sciences, also in the directorate for computer and information science and engineering. And in addition, there are what are called convergence accelerator programs that consider projects, a large component of which could involve synthetic biology. Nevertheless, there's no time really for me to go through those things. But what I would like to bring to your attention is another aspect of the funding opportunities of NSF. And in general, we are provided with several mechanisms that would enable us to advance science and engineering, and in particular for synthetic biology. And there are two examples I would mention. The first is SynBYSS, which you might remember, that is the Synthetic Biology Research Center. And SynBYSS was a multi-university research community that helped to lay the foundation for synthetic biology here in the US by making some very important seminal contributions in initiating networking among research labs in the scientific area. Well, the progeny organization, the EBRC, Engineering Biology Research Consortium, which is an inclusive community of scientists that are committed to the advancement of synthetic biology in the context of national and global needs. I am the program manager for that project, right? And these two examples are just meant to illustrate to you that NSF, in addition to the research type projects, we also fund projects that do not involve research, but would enable community advancement in various ways. And for the EBRC and SynBYSS, we have funded these organizations to the tune of millions of dollars. So this leads to the point that I'd like to make before signing off here. I bring to your attention the little known fact about NSF, and that is that there is much flexibility in the types of projects we accept and the manner in which we review and fund these projects. Therefore, if you're thinking about submitting proposals and making applications to us, consider thinking creatively and outside of the box, so to speak. we're always looking for ideas that are transformational, not only in research, but also in the impact that these projects may have on our society. Therefore, when you're thinking about NSF, I would encourage you to consider approaching us with your non-traditional ideas that are bold and would potentially make a significant difference to the areas in which you are conducting your synthetic biology research. And if you have to collaborate for success, we do support collaborations, and there are special programs that would facilitate international collaborations. So if you have international partners that you would like to connect with in order to conduct or to add more dimensions to your research program, you could consider looking at these programs, these types of programs within NSF. Now, you may have heard that NSF will have, well, no, NSF may have a new directorate because the president has made a request that in the president's request for NSF, there are plans for adding a new directorate called the Directorate for Technology and Innovation Partnerships, in which would add more than $800 million to the NSF budget, and no doubt will offer tremendous opportunities to you to submit your ideas in synthetic biology. So in closing, I would say that it is clear that biotechnology will form the foundation of future economic growth. And as you position synthetic biology to meet the challenges of the 21st century, both technical and as well, you would need to consider those issues that are associated with public understanding of the scientific area. But when you are developing your research plans, I would like to encourage you to think expansively and holistically about what you want to achieve and how you would like to go about achieving your goals. And so with that, I shall turn the floor over back to Taisuk to continue this exciting program.
Oh my God, wow. Amazing talk and inspiring insights. And also thank you for introduction of NSF and funding opportunity. Also, thanks for giving us the insight again into proposal preparation, especially for young people who really wanted to know what to do in terms of those kind of things. Also, I mean, I really thank you for NSF. I mean, continue support SynBYSS and EBRC. Myself, I was a SynBYSS kid for entire my training career from PhD to postdoc, and I now one of the council member in EBRC, and you mentioned, and I believe the US is leading the SynBio community thanks to your support for SynBYSS and EBRC. So thank you so much for continuing to support.
Sure. We're always happy to be able to be involved in organizations that would advance scientific areas. Yeah.
Thank you so much. So also I 100% agree with you. Sustainability is very important, and I want to say, or also I want to see our city looks like your background, picture with the beautiful nature and the protected city. Okay, thank you so much. I love it.
My pleasure. It was my pleasure. Thanks for the invitation.
Thank you. Okay, so now the main speaker of today with a little bit longer introduction. So Dr. Christian Cuba-Samaniego is a postdoctoral scholar at UCLA, working with Professor Elisa Franco. His work lies at the intersection of feedback control and mammalian synthetic biology. Of course, very different area, but combining two things together and for the fascinating application, including therapeutic applications. Christian's research goal is to design and integrate feedback control and pattern recognition paradigms for biotechnological applications. His passion for programming and circuit design started in his undergraduate study in Lima, Peru, where he focused on building and programming self-driving drones as a mechatronics engineer. Inspired by control engineers working in biology, he joined the PhD program at UCLA under her guidance, the Elisa, and his graduate work centered on mathematical modeling at the intersection of dynamical systems and control theory to understand the behavior of biological networks, and result in 11 journal and 15 peer-reviewed conference papers. To complement his theoretical background with experiment, he recently joined Ron Weiss's lab at MIT as a postdoctoral researcher focusing on mammalian synthetic biology. Today, he will share his first experimental work on building synthetic networks capable of complex decision-making machine learning networks in living cells. I'm so excited, seeing this kind of combined effort, experiment, and of course, the theory and the computation. And I'm excited to see, and I cannot wait. And then so, Christian, take it away. It's all yours.
Thank you for a nice introduction and a great opportunity to share our science with everyone. I will start sharing my screen. Okay, awesome. As Ty mentioned, my background is in engineering, and I'm going to present my first experimental work. When I started this project, I did not have any experimental skills at all. And the journey was very challenging to learn all the molecular biology techniques. At the same time, it has offered me opportunity to connect with different mentors who taught me how to do biology and appreciate even more how much work it takes to push an experimental project. So the current project I'm going to talk today, it is the main work during my postdoctoral research at MIT with Ron Weiss. And it consists on how can we engineer synthetic network capable to process multiple input of biomolecular patterns. So let me motivate the problem here. So engineering biology has like a great potentials to power our world, fuel our planet, and feed us. And so in particular now we can engineer, for example, our own T cells and program them with capabilities to recognize, let's say, cancer cells from normal cells. Here in this picture, in this recently paper, they have characterized the patterns of antigens that we can use to distinguish a normal cell from a cancer cells, which are represented with the blue and red dots. And what they did was they characterized a lot of the level of expressions of these antigens in different organs with different patients, and trying to identify what are those biomarkers associated with specific cancer cells. Those experimental data are very interesting because it gives us an idea what are the patterns associated for each disease, and more importantly, what kind of computations do we need in order to engineer the cells. Now, nowadays, current technology are driven by logic-based operators to engineer these cells for therapeutic applications. For instance, CAR T cells. Here you have a simple schematic of a logic-based design where a cancer cells can be identified if these exposed to antigens, and if one of them is missing, then the T cells will not kill the cell. However, this approach requires and demands that the biomarkers to be compatible with a logic-based operators, and therefore it can be very challenging to understand more complex behavior. For example, here in this schematic that shows for two different antigens, the black dashed lines would correspond to an ideal and gate operator. As we can see, there are a lot of red dots outside this box. So then this requires that we need to improve our computations in order to increase the efficiency of our therapeutic applications. For example, maybe a decision boundary shown in the green line could have a better performance, or we could even require a non-linear decision boundary in the circuit overall. So then artificial neural networks are really great for classification problems. If we abstract these problems, we can represent different type of pattern recognitions, where in the left, it shows a linear decision boundary, which means we can separate the gray and green dots with a line. On the right side, we have a non-linear decision boundary, which in this case will require a more complex or two lines in order to separate the orange dots from the gray dots. So then artificial neural networks, to give you a little background, consist of the interconnections of multiple nodes, where each node can process the sum weight of the inputs and feed it into a function. Here you have a simple abstraction of these functions, and the most popular activation functions, or the most common ones, are these linear rectifier or sigmoidal type ones. So then our goal is to bring artificial neural networks into cells. However, there is no way that we can shove a GPU inside of these cells. Therefore, we require to implement such computation using the language of biology. Of course, there is a lot of preliminary work on this and this type of problem. And I think there is a lot of progress in vitro designs of these artificial neural networks that relies on DNA-based technology. Here you have two of many examples that they have been used to implement linear classifiers to distinguish letters, numbers, and also it can be used for DNA computation. On the other hand, there is also a lot of evidence that biology can indeed compute these type of neural computations using genetic regulatory networks, or in this other recent paper where you can see the interaction of receptor of ligand can produce different type of patterns that can drive endogenous process. So what I'm going to talk today is I'm going to introduce and analyze how we can use molecular sequestration to realize this type of chemical neural networks in living cells. Then I will explore what will be the ways associated with this network. How can we characterize a library of these particular set of chemical reactions experimentally, and how we can compose a multi-layers. Just to give you some challenge here is when we connect these neural networks, the weights can take positive and negative values. Now, all the species concentrations or kinetic rates in biology, they are positive. So therefore, how can we incorporate these virtually negative values, because that will expand the capability to engineer different decision boundaries in cell. So now, let's start. Molecular sequestration consists of two species, Y and zeta, where they can form a mega complex that does not interact with the reaction overall. So For example, if you have 100 molecules of Y and 20 molecules of zeta, and then this will result as producing 20 molecules of complex and 80 molecules of Y. We can think this as a subtraction operations if the sequestration rate is very, very fast. Based on that idea, we can use the sterility analysis or the equilibrium of the systems and show that indeed the output Y is going to be proportional to the difference of the production rates divided by the degradation. So then if we explore the input-output for a constant value of input V that produce the species zeta, we can see this as a thresholding mechanisms. And if we change the value of this thresholding, the value of V, we can indeed change the thresholding of the input-output behavior. So then if we plot the maps of inputs and output for U and V, we can indeed observe this linear decision boundary that happens in the diagonal of this square. And now we're going to take advantage that because these mechanisms could potentially realize subtraction in this particular regime, we can take advantage of this to incorporate negative values due to the subtraction operation. Then if we process multiple inputs that produce the species Y and zeta, we can rewrite all this expression as the sum weight of inputs where the weights will be associated with the production rates divided by the degradations. And this can also incorporate the positive or negative weights. Any input that produces the species Y, which is the output of your systems, will be associated with a positive weight, and any input that produces species zeta will be associated with a negative weight. So then therefore, we can make even an abstraction, an analogy, and an ideal perceptron in machine learning, which is a process the sum weight of inputs and feed it into a linear activation function. So then we can see here that this mathematical analysis can indeed show that there is a lot of, that we can use these systems as a single fundamental unit of perceptron using chemical reactions. So we have published our theoretical work with Andrew and perform all this analysis. Now, let's move to the experimental part of this. We show you that molecular sequestrations can indeed implement this fundamental perceptron. So then here we explore two first realizations, which consists of the Endoribonucleases. ERNs are proteins that they combine to a specific sequence of RNAs and perform a cliff of the RNA molecule. In addition to that, it remains bound to the target or the hairpin of the RNA. So then here, Bree in the waste lab at the center, she has been characterizing a lot of these systems with different proteins. And here we have like a large library of candidates that implement sequestrations that we can use to implement these molecular perceptron nodes. So now that I have shown you with mathematics on how we can abstract, how can we abstract the chemical reaction into an abstract node, now let's start from the other way. Let's start from an abstract representation of a single node that consists of two inputs that are associated with a positive and negative weight, and let's uncover and let's see how we can translate this into a chemical set of reactions. So then to do this, we need to incorporate a molecular sequestration mechanisms through the interaction of Cas-E, the Endoribonuclease protein, that it will target the mRNA. Then if we encode this into genes, we can measure the input-output of the systems if the mRNA translates into a protein, in this case, EYFP. But here what is missing is how can we track the input-output of the systems? So to do that, we use markers, other fluorescent proteins that it will be contransfected with EYFP and Cas-E. So to do this, we're going to use a technique called poly-transfection, which was also developed in the waste lab. And I will explain you next what it is. So normally when we transfect our synthetic circuits, we put our plasmids into a transfected reaction. and deliver it to the cells. Every cells will adhere different copy number of plasmids. And here you have a schematic of the distributions of fluorescence. And here you have an experimental characterization of this. So now if we combine two different plasmids, the ratio of these will remain the same in all the cells, but the copy number of plasmids can vary. And we can see here that in this experimental data, where each different color will correspond a different ratio between the plasmid in blue and red. And we can observe that it only covers partially the parameter space of this system. So then what my colleagues discovered was that if we transfect the plasmids separately, this is the poly-transfections, in a single experiment, we can show that the cells will have a different copy numbers of the plasmids in blue and red, which means they will not be correlated as in the case of co-transfection. And therefore, in a single experiment, we can cover all this parameter space for two plasmids. Using that idea is where we can use to characterize the input-output of our circuits here. So this work has been driven by 3 colleagues in the Waste Lab, Jeremy, Bree, and Ross, and I recommend you to check this paper and to use it for circuit characterization. Now, going back to the characterization of our circuits, the plasmids that are inside the blue and the red color will be co-transfected. So in that way, if we measure the fluorescent protein of tag BFP, indirectly we will be measuring the relative copy number of the plasmid EYFP. Similar, if we measure IRFP, we will measure the copy number of the approximate copy number input of the plasmid Cas-E. So then by measuring just the fluorescence, we can cover the input-output of the systems. Then by measuring the output of EYFP, we can characterize the overall behavior of the circuit. So here you have one of many representations of these circuits where in the x-axis, we vary the total amount of molecules of EYFP. In the y-axis, we vary the level of expressions of the Cas protein. As in the simple model predicted, that the sequestration mechanism will show us a kind of a thresholding effect with a decision boundary close to the diagonal. The experimental data showed similar trends overall. Then if we analyze one section of these input-output, for instance, for a constant value of Cas-E, we can see that there is a regime where there is no level of expressions, and then we have a linear expression level of our proteins. So now, our simple model also suggests that the production rates divided by degradations will tune the weight on the system. So in order to verify this hypothesis, to the same circuit I just described you before, we're going to change regulatory elements that help us to tune transcriptions, translations, and even degradations of the proteins, which is shown in the left. So first, we're going to change the translation rate of the Cas-E protein. So in this case, we're going to use a UROFs, which are repeated sequence that are going to control the translation rate. Like in this case, we start with basically a non-UROFs at all. And then we can basically observe that we are expressing more Cas-E, therefore, we are going to reduce down the level of expression of the system. But now, the moment we start decreasing the level of expressions of Cas-E or of their translation rate, we can start turning or changing that decision boundary, the linear decision boundary that we had before. And if we even put it down, the translation rate, we can even rotate completely the decision boundary of our system. Now, this is tuning the translation rate, but we can also tune, for instance, the copy number of the plasmid, the ratio, and this indirectly has an effect of changing the transcription rate. And here we can observe similar effect that we can indeed tune this decision boundary. Similar, we can use degradation domain proteins to tune the decision boundary of our systems. And all of these experiments kind of like agrees with the simple model of how we can tune this linear decision boundary experimentally. Now, how can we use this linear decision boundary for applications? How can we process biomarkers? At the beginning of this talk, I introduced the problem using antigens, but we can also, for instance, process internal biomarkers such as microRNAs. Here in this schematic, we have level of expressions of microRNAs between hex cells and HeLa cells. And here we can use, for instance, the microRNA 21 and lead-78 to kind of like classify or differentiate HeLa cells from hex cells. So just to illustrate, we could, for instance, try to implement a linear decision boundary shown in green. So then to do this, we would require a single node that process the two inputs, X and X2, which will be the microRNAs, and then X0 will represent a bias in the system. Like in this case, the weights for the inputs are negative and the bias is associated with a positive weight. So then transferring these abstractions into a synthetic circuit, it will consist of two different genes producing Cas-E that are associated with a negative weight, and the output, which process the bias in the systems, will be associated with a positive weight. So now to incorporate the biomarkers as inputs, we add some sequence that the microRNA can target in order to degrade the mRNA of the system. So now, because the same experiments of poly-transfections can be used to optimize the circuit, like in this case, by changing the copy number of the plasmids, is indirectly changing the weight of the circuit because it can either change transcription or translation rate. So then we can use this kind of like approach to basically optimize what will be the more optimal ratio between the orange, red, and blue in order to have a good classification for HeLa and hex cells. So then here, once we optimize the circuits, we can indeed show that for HeLa cells, the system will perform an output that we can measure the fluorescence, and for hex cells, it will not show a fluorescence at all. So, and then we can even perform that, we can characterize the accuracy, the sensitivity, and specificity of these microRNA classifiers. Now, in order to build a more larger circuits, we need to expand our library of sequestration mechanisms to implement these single perceptron nodes. So now we're going to explore how can we achieve similar behavior using proteins. So using the simple schematic as before that consists of positive, negative weights are associated with the inputs, now we're going to explore on how dimers of how we can use dimers to implement these sequestration mechanisms for heterodimers. For example, TED-R is a well-known established systems that it can be used for either activation of repressors in mammalian biology. So now when we create like mutants of TED-R that it can affect, for instance, that it can recognize different DNA binding domain, but maintain the core of dimerization the same, this can indeed incorporate the sequestrations in the systems and form this heterodimer complex. We can expand and then to check of this hypothesis, we perform the following circuit. So we create a TED-R mutant that it will not bind to the target's sequence of the TED-R repressor and to evaluate their input-output behavior. Similarly, we can use these for any other proteins. For example, recombinases. Recombinases are great proteins because it basically gives you an on and off response comparing with other systems. And because there are some particular recombinases that they can reverse the flipping of the target, and we can basically re-engineer the systems to implement sequestrations and therefore have the advantage of this thresholding effect overall. So then when we perform the characterization of those circuits, we can indeed observe that there is this linear decision boundary, although now the activation type of function is not a linear rectifier, it's a different type of function. It still maintains this linear thresholding effect that we can use to engineer these biomolecular neural networks in cells. So now that we have shown you how we can realize a single node that for a biomolecular neural network, we have also shown you how different ways to implement it. Now we're ready to implement and compose a larger set of circuits. Before going there, we performed some mathematical analysis in terms if we connect a feed-forward network based on sequestrations, we would like to guarantee that the systems is going to be stable. For example, it will not show oscillatory behavior or bistable behavior. And that means that we can use our steady state analysis in order to predict the input-output behavior of the system. So let's now go to the next example where we're going to aim to build a nonlinear classification, like in this case, a bandpass classifiers. So the circuit consists of two nodes. The first node is processing the inputs X1 and X2 associated with positive weight, and it also has a bias with a negative weight. Similar as before, we will use for the first layer the sequestration between mRNA and protein, in this case CSY4, which work in a similar way as Cas-E protein. Now, this mRNA can produce, in this case, a protein for the second layer, which process the same inputs X and Y associated with the positive weights, and then it has a bias with a negative weight. And now the interconnection of the first and the second node is associated with a negative weight. So now to realize the second way, we're going to use a protein-protein interaction, where the interconnection of the first node and the second node will be when mRNA can produce a recombinase proteins that it will interact for the second node. So now the system, although has more elements, we can still be able to characterize the circuit overall using this poly-transfection. So here we use fluorescent proteins to keep track of the input in the systems, and all the plasmids inside each color, red, orange, and blue, will be transfected in a single transfection experiment. So then when we perform our simulations to try to understand and to tune the weights of the systems in order to deliver this bandpass classification, we use these to guide experimental characterizations before their optimization. And here you have one of the examples that is optimized, where we can indeed observe these bandpass classifiers. But more importantly, we can, by changing the weights of the systems, we can change this decision boundary, as I have shown you before with a single element. So then, based on the demand of the patterns, we can achieve a more complicated decision boundary tailored for each application. Now, further, now we test that if we can still predict the input-output behavior if we increase the complexity of the circuit. So now we're going to explore a 3-node circuits that it can achieve a nonlinear decision boundary. Like in this case, a dual region pattern recognition, which is basically the opposite of the dual region. So of the bandpass. So then, similar as before, for the first layer, we're going to realize we're going to use mRNA and protein sequestration, and for the third node, we're going to use protein-protein sequestration. Without going into much details, but I just want you to see that all the plasmids that are in the same color, it will be co-transfected, and therefore we can use this poly-transfection technique to characterize the circuit in a single experiment. Similar as before, we use simulations to try to understand what would be the ratio patterns that we need to perform this nonlinear decision boundary. And here you have some of our preliminary experimental data where indeed we can see this dual region pattern classification in the circuit. One great advantage of using mRNA protein and protein-protein is that we skip the need to use an intermediate transcription factors between them. So we can implement 2 layers without an additional transcription factors. Now, if we would like to implement a third layer, for instance, now we will require to use a transcription factor in order to scale this circuit larger. So in summary, we have shown you a platform how we can achieve these biomolecular neural networks for pattern recognition using molecular classifiers. We have identified how molecular sequestration can indeed implement this linear thresholding response. We can use this linear decision boundary to perform more complex systems. Also, we have characterized a library of them that we can experimentally use to build larger complex. And finally, composing multiple of those networks, we can implement more complicated pattern recognitions that it can be tailored based on their application. So another exciting work that is coming from this is similar as we study molecular sequestration, we also study signaling-based networks. And here in this other theoretical paper, we make similar analogy on how signaling networks can process some weight of inputs associated with positive and negative weights. At the same time, lucky for us, my colleague, Ross Jonas in the Waste Lab, he has been characterizing these systems, and here you have some of the experiments that indeed they show this linear decision boundary, which is very promising for the experimental validation of our theoretical paper. Next, we're also exploring how can we implement learning in the circuit overall. Since my background is in a control engineering background, I'm trying to address this problem on designing an adaptive controller for optimization. Once we're able to achieve optimization using chemical reactions, we can reuse this control strategy to realize learning to basically optimize these circuits inside the cells. So I would like to acknowledge again, this is a lot of progressive work from the Waste Lab. And I have a lot of great colleagues with Andrew, Jose, and Nicolas that they have been helping me a lot in learning the biology and being supported in pushing this project. And there is a lot of people that have contributed a lot, as well as Elisa Franco, who has been supporting to continue in this work and now in my current position. And then a lot of getting rid of feedback on trying to improve these systems overall. Before finishing this, I would like to advertise. Now, the Cold Spring Harbor Laboratory has the summer course. In this case, there is a synthetic biology course with great instructors. We have a bunch of great speakers this year. So I would strongly recommend you to apply it. There are fellowships, and it's a really nice experience where you will interact during 2 weeks with the speakers and do a lot of science in a very, very nice environment. So I invite everyone to apply it and spread the word about this. Now, if you have any questions, I will be happy to take them.
Wonderful. Wow. That's amazing. Amazing work and the fantastic talk. And also, yes, I mean, Cold Spring Harbor, SynBio, if you are young and then passionate about synthetic biology, apply. I believe that submission deadline is coming soon, right?
Yes, it's on April 1st.
Right. I remember. So that's fantastic instructors and all the great speakers. I mean, you cannot have those kind of speakers in one place. I mean, so great. So somebody just said, I mean, can you put the link for the course? I mean, can you do that? I mean, while I'm kind of looking for the question here.
Just 1 second.
Yeah.
Let me. Oh, yes, I will put the link shortly.
Okay, so this is wonderful. So any question, Dave, you have any question or comment?
Well, I would just like to say it's a very fascinating talk. Yeah, I would just have one little question. How can these circuits be implemented, what kinds of applications they would have, specifically if you want to be interrogating, you know, cellular control or cellular regulation. Do they have any kind of application in that manner? Or, you know, are they flexible enough to be used like that? Yeah.
Sorry, could you repeat that question one more time?
Yeah, I was wondering if these systems that you have devised here, if these are the type of systems that could be used that could be, you know, implemented in cells or adopted in any way so that it could be used to interrogate cellular processes, like understand how cells work, understand how cells are regulated. Are these the type of circuits that can do that sort of thing? Or if not, what are the kinds of applications you would see for these? Yeah.
I see. That's a great question. So I think in order to answer this, we can talk about two categories. For example, the first one could be engineering synthetic circuits. As I start with example of CAR T cells, if we use the biomarkers of antigens as biomarkers to basically engineer when it can recognize a cancer cell from a healthy one, that's, I think, one of like immediate application of this. But it's not only limited to that. It's based on what kind of biomarkers is the systems. This platform can easily be adapted to process the, different patterns behavior. For example, we can use in the case of microRNAs, we can use transcription factors, for example, to steer development programs. Right, I think that the opportunities are a lot in terms of identifying the biomarkers. Now, for the other side is, how can we use this to learn the biology? Is biology performing this type of neuromorphic computation? So then there are some very interesting papers that I think I didn't spend that much time, but here, where basically, for instance, when we have the receptor ligands. So in this great paper, they actually explore these combinatorial complex formations. From our perspective, is basically, oh, you have sequestrations everywhere in this type of system. So then this could help us to basically understand how nature is performing endogenously in many of those endogenous processes. Although it's not very similar right now in the sense that in our circuits, we isolate each sequestration unit, but endogenously, you have this crosstalk of sequestrations. So then that kind of like performs, okay, what additional function is that embedded in biology? Yes, I think it's a very exciting, and it can like give us like a different perspective on how nature may work as well. We can go in both ways, engineering synthetic circuits and understanding the endogenous circuits as well.
So amazing. So any question from audience? Okay, in this case, I'll ask the challenging question. So as you know, I am very fascinated by living computer concept or ideas, but we still have many challenges to overcome to realize this computer, the living computer kind of idea. So could you comment on the current challenges, remaining challenges, even though we have lots of, many years of achievement, and then remaining opportunities. Could you comment on that point?
Yes, so as we mentioned before, currently we're using logic-based approach to implement our computations. And it's in the case that I just show here that sometimes this is limit or this requires that the systems to be compatible with a logic-based design. So then we need a different strategies. One way to address this challenge is to using basically analog computing, or in particular is machine learning for pattern recognition. So now the challenge that we will have now is, okay, how can we identify what other mechanisms we can use that it can basically realize these positive and negative weights in the systems. Implementing negative weight is a key because it allows us to create different decision boundaries. Second challenge, I think, is the optimization of these circuits. And I kind of like the idea of what the Boyle Lab is doing right now. Recently, they published the second version of Cello, which means, can we standardize the parts to implement these molecular pattern recognitions, where we don't need to perform the characterization, but rather saying, if I would like a linear decision boundary with a specific angle, so then I could just take the part that's being characterized rather than optimizing the circuits. And another challenge that I envision with this is that very often the circuits that we perform, they deliver a suboptimal performance. So then that's kind of require like an optimization of these circuits in vivo, that it can actually meet the requirements for application. And I think that's going to be like the challenge here, that how can we automate this process? It's a problem that we have been working as a strategy, how we can accelerate this process. So then we kind of like, it will be nice to have like a platform that everyone can use, that it can democratize the optimization, because right now, optimization of synthetic circuits are basically limited to big companies like Ginkgo's, for instance, where you have these automated infrastructures to optimize your circuits. And that is very limited for academic or small labs. So then in order to push these technologies, and not only to realize these molecular pattern recognitions, but in general, for any synthetic circuit design, we need to push the optimization of our circuits that is accessible for everyone.
Okay, fantastic. Thank you for your amazing answer for my challenging question. So any other question? Okay. So thank you, Dave. I mean, for your time. I mean, I love to see you somewhere else in person. Thank you so much.
All right, thank you for the opportunity.
Okay, so I want to close in that case. Okay, so thank all for joining and staying today. We will meet again next week on March 3rd. Thursday, the same time, the same Zoom link. We'll have Professor Matt Drisa from Cornell University, the director of Cornell Institute of Biotechnology, and Josh Tico from Stanford. As usual, the follow-up informal chat will occur without recording, and please stay here if you are interested in chatting with today's speaker and me. And I'll promote you to panelists who can speak and show your handsome and pretty face. So thank you so much, and I will stop recording now. Okay.